Our next speaker is Mike Boxem from the University of Utrecht, and he's going to talk about mapping the C. elegans polarity interactome.
Thank you very much. So I'm going to switch away from transcriptional regulation to protein-protein interactions, and also probably slightly more modest approach. We're not trying to understand the entire worm, we're just trying to understand how cells polarize. So what I want to tell you about is our efforts to better understand the network of molecular interactions that underlies cell polarity. And when I say we, I mostly mean a graduate student who graduated last year and is now a postdoc at Mass General Hospital, Thijs Koorman, who did the bulk of the work. Yes, there we go. Okay, so as most of you will be aware, cell polarity is an essential feature of cells and an important part of development, regulating processes from asymmetric cell division to cell migration, and also the formation of distinct membrane domains in cells like epithelial cells, as shown here, or neurons. And over the past decades, work in C. elegans, of course, from the Kempfri's lab and many others, and in Drosophila has identified a number of key regulators that are involved in the regulation of cell polarity. And probably one of the best understood systems of polarization is the epithelial cell, which I'm showing here, where opposing actions of apically localized cortical proteins, the Crum's complex and the PAR complex, and basolateral proteins, namely the Scribble complex, generate an apical basal axis of polarity and two distinct domains. But even here, we don't understand all of the functional interactions between these proteins, and they can also differ between organisms and cell types. So, for example, we and others have studied the Crum's complex, and this is a protein complex that's essential for cell polarity establishment in several epithelia and Drosophila, but in C. elegans by itself, it's not essential for cell polarity. And the same actually goes for the LGL protein. And what's more, we know relatively little about how this cortical regulation of cell polarity integrates with other aspects of cellular polarity, such as establishment of a polarized trafficking machinery, organization of the actomyosin cytoskeleton, such as, for example, the polarization of microtubules that you saw earlier today. And so ultimately, to get a full understanding of cell polarity, we also need to have a comprehensive overview of the protein-protein interactions that are involved. So with that in mind, we set out to identify the protein-protein interactions underlying cell polarity, and we do that in our group using the yeast hybrid system. And then to begin to identify which of the interactions that we identify are physiologically relevant, we combine this with phenotypic mapping of the proteins in the interaction network using different models of cell polarity in C. elegans. And the idea is, of course, that by combining these two methods, we get a better sense of which protein interactions are actually involved in polarity establishment. Okay, so as I mentioned, we do this by yeast hybrid, and we performed screens using a series of 69 polarity regulators as baits. And these are proteins that are either known to be essential for cell polarity in C. elegans, or that are homologs of polarity regulators in other organisms. And for each of these proteins, we used a full-length construct as a bait construct in the two-hybrid system. But in addition, we screened for each protein a number of different fragments, fragments that were designed based on the domain composition of the protein, as indicated in this example here. Get the pointer to work, yes. And the reason we do this is twofold, really. The first is that we're interested in also identifying not only what are the proteins that interact, but what are the domains that mediate these interactions. And the second is that our previous work had shown that by using a fragment-based approach, you can increase the completeness of these interaction networks. And that's just for technical reasons that a full-length fusion construct that you express in yeast isn't always functional. So we screened these fragments and full-length constructs against two libraries, a traditional cDNA library that might contain all C. elegans genes, but probably contains 15,000 or so, and a fragment library that was designed specifically for the identification of interaction domains, but it only contains fragments of 750 proteins that are involved in early embryonic development. In these screens, we identified, as you usually do, a large protein interaction network containing 439 interactions between some 300 proteins. And what we noticed immediately was that most of these interactions were indeed found only through the use of additional fragments as baits. And quite a few of these extra interactions are well-described literature interactions. So that indicates that we indeed increased the completeness of the network through this approach. So the other reason that we used fragments was the identification of interaction domains. And I just want to show you a few illustrations. So these are interactions that are well described in the literature that we then recovered also in the yeast hybrid, and for which also interaction domain information was known. So for example, here I'm showing you the full-length par6 protein. And in yellow, I'm indicating the region that our two hybrid experiments say is responsible for the interaction with PKC3. And in blue, I'm showing you the domain that was shown to be essential for this interaction in the literature. So that's well known that the PB1 domain of par6 mediates the interaction with PKC3. And these are two other examples. And you can see that we are nearly exactly replicating the information in the literature. That's not always the case. There are also cases, of course, where our approach, which is a broad and systematic approach, is not able to map an interaction domain as finely as a dedicated approach in the literature, and then you'll find that the literature domain is much smaller than what we uncovered. But the important thing is that in almost all cases, something like over 95% of the cases, the interaction domain that we identify contains the domain that's described in the literature. So the domains appear to be of very high quality. And speaking of quality, we also validated the overall quality of the protein interaction network. And we did that using two orthogonal approaches. We retested a small number of interactions using affinity purification from mammalian tissue culture cells. And we were able to recover about half of the interactions that we tested. And from our previous work, we know that that's actually a very good number for retesting a set of protein interactions in a different orthogonal assay. And with the help of the group of Jan Tavernier, we also tested a much larger set of interactions in a mammalian protein-protein interaction system that they developed called MAPIT. And the advantage of this system is that it had been benchmarked very well against previous data sets, including a set of interactions curated from the literature and a set of random proteins that serve as a negative control, because just selecting proteins at random, you don't expect them to interact. And what I'm showing here is the retest rate at a stringency level of the assay where random pairs show no retest, none of them test positively. And you can see that our interaction network retests at a similar rate as literature-curated interactions and as a previously generated C. elegans protein interaction data set. Okay, so from all of that, it seems that we have now a large network of protein interactions that appears to be of good quality. But the drawback of the two-hybrid system is always that a protein pair that can interact in yeast isn't necessarily a protein interaction that is relevant in vivo. And to begin to select those pairs that are more likely to be biologically relevant, we followed up our protein interaction mapping with a systematic phenotypic analysis. So what we did was inactivate genes corresponding to proteins in the network by RNAi and examining their phenotypic effects using different markers of cell polarity. And we used a number of different markers, some more traditional markers highlighting cell polarity, for example, in epithelia like the intestine or the seam cells, or a par polarity in the embryo. And we also used some less frequently used markers examining, for example, yolk trafficking from the gut to the oocyte, which is a process that's dependent on the par proteins, and establishment of axon versus dendrite identity in the PVD neuron. So here you can see only the axon, but I'm sure all of you have seen these beautiful candelabra structures from Benjamin Podbilevich's lab. But this is a marker that localizes specifically to the axon. So what we did was we first tested each of the 69 bait proteins in all of these different strains. And we scored them on a scale of 0 to 5 based on the severity of the phenotype. And to get an idea of how well the RNAi phenotypic signatures that we get match known functional associations, we used this information to perform unbiased clustering. And it was very gratifying to see immediately clusters coming out of proteins that are known to interact together, such as the PAR proteins, or the junctional components AGM1 and this large, or the Lin2, 7, and 10 complex. And next, we performed phenotypic screens of the bait proteins, so the interacting proteins of a particular bait, but only in a strain where there was a phenotype. So if a bait protein scored a phenotype in 1 or 2 strains, then we only tested the praise in those strains. And all combined, we identified 100 protein pairs that we knew already physically interact, but that where RNAi results in defects in the same polarity-related process. So just to show you an example, we identified an interaction between this large and a protein LST1, which affect both RNAi for both of them affects the intestine, resulting in a defective morphology, as shown here by this apical marker, PEPT1. And they also result in defects in the normally apical clustering of recycling endosomes marked by RAB11 vesicles. RAB11 GFP. But not all phenotypes are always overlapping very nicely, so we have quite a few cases where RNAi against 2 interaction partners affect the same tissue or process, but in distinct ways. So for example, inactivating ERM1, we occasionally get multinucleated seam cells, and if we inactivate this uncharacterized interaction partner, we get a sort of spiky appearance of the network in these gap-like structures between seam cells. But nevertheless, the fact that they affect the same process is an indication that that these interactions are physiologically relevant. And to examine a little bit more systematically if pairs that show an overlap in phenotype are predictive of a true functional association, we compared our data with Go terms, similarity, and with prior publication. And we saw, for example, that protein pairs that interact and show an overlap in phenotype are more likely to share a similar Go term, and they are less likely to share a non-similar Go term. And we also found that 9 out of the 100 protein pairs that show an overlap in phenotype had been prior published in single protein studies, whereas only 5 out of the remaining protein interactions have been previously published. Okay, so all of that indicates that we are indeed on the way toward predicting which proteins are physiologically relevant. And of course, we only use a subset of markers. So I hope that this is can be used now by the community, not just C. elegans researchers, of course, as a resource to have sort of a set of candidate proteins to examine in your favorite system of polarity. And I'd like to spend the last few minutes of my talk on telling you about one particular protein interaction that we examined in more detail. And that's a protein interaction between the row gap protein PAC1 and the cortical polarity regulator PAR6. And the reason this initially caught our attention was that the phenotypes of these proteins by RNAi overlap significantly. And also that quite a bit was known about PAC1. So studies from Jeremy Nance's group had shown that PAC1 is required for radial polarization of the embryo. So here you see localization of PAR6 and PAR3 and PKC3 have the same pattern. In the 6 to 8 cell embryo, start to localize to the outside of the embryo, to these contact-free cell surfaces. In a PAC1 mutant, PAR6 decorates the entire cortex. Now, PAC1 itself localizes to the sites of cell-cell contact, and more recent work from Jeremy's group has shown that this is through binding to E-cadherin through an intermediate adapter protein. So, as I said, PAC1 is a row gap protein, and it specifically inactivates CDC42. And CDC42 is required for recruitment of PAR6 to the membrane. So the model is that PAC1 locally inactivates CDC42, preventing PAR6 from being recruited to the sites of cell-cell contact. So as you can see, PAR6 and PAC1 don't overlap in localization. and nevertheless we found a physical interaction. So we wondered, is this physical association relevant? And we worked with Jeremy Nance's group to investigate this further. We first mapped the PAC1 par6 interaction domains. Our yeast-to-hybrid data told us already, and we refined this a little bit manually, that the PDC domain of par6 binds to PAC1, and it binds to this region just downstream of the row gap domain of PAC1. So this is a case where PDC binds an internal site on the protein. Often it binds to the C-terminus. That's not the case here. We confirmed these interaction domains by affinity purification from tissue culture cells. And then Jeremy's group, Diana Klomstra, graduate student in this group, generated a deletion mutant or a deletion construct of PAC1 lacking the par6 binding domain. And then she investigated, can PAC1 lacking the par6 binding domain still mediate radial polarization of par6? So here is just again the wild-type localization of par6 and the localization to the entire membrane in a PAC1 mutant. Jeremy's group had already shown previously that expressing a GFP-tagged PAC1 in this mutant background can restore radial polarization of par6. And here you see localization of this GFP fusion protein. But when you express GFP-tagged PAC1 lacking the par6 binding domain, this construct can no longer restore or can no longer mediate radial polarization of par6. Here you see that the construct is still localized to the right location, to the sites of cell-cell contact. We also quantified expression levels, so this is not due to differences in expression levels. So what this data says is that this interaction is in fact biologically relevant, and that in addition to the gap activity of PAC1, binding to par6 is essential for radial polarization of par6. So with that, I'd like to finish. I forgot to put in a slide that we generated a very nice website where you can browse all this data, but you can find the web address in our paper. It accumulates not just the data that I showed you today, but also all previously known C. elegans interactions that we curated from various databases. So as I said, most of the work was done by Thijs Koorman, and also with significant help by a former postdoc, Monique van der Voet, who is talking here today about her current work on Drosophila, and help from graduate student Joao Ramalho, and our collaborators Diana Klomstra in Jeremy's group and Irma Lemmens in Jan Tavernier's group. And thank you for your attention. Sure.
So what do you think this interaction is for? So could it be that par6 regulates the gap activity of PAC1, or any idea on that?
Yeah, we've speculated back and forth about this. I think one of the things that it could be is that, for example, that you might want to trigger maybe specifically the CDC42 pool that's bound to par6, and that might be helped by PAC1 only recruiting that particular pool. Something like that is our best guess, but we don't have a definitive answer for a mechanism there. No.
There's another question?
And Mike, that was really nice. I would just want to make sure I caught one thing you said. You retested literature interactions, and it looked like you said only 25% of them retested.
Yeah, so this is that's in the MAPIT system. This is actually a general problem. So you will hear a lot about false positives, but false negatives are also a very big issue in the protein interaction mapping field. So whenever you take a set of well-curated literature interactions, and so the Vidal group has done this, for example, with 5 different assays, you'll find some interactions that are easy to work with. They will test positive in any system. And then you find other interactions that will only work in 1 or 2 systems due to post-translational modifications, etc. The MAPIT you can do, I showed you bars at a single cutoff. You can sort of have a scale of different cutoff levels, but as you go lower in the cutoff level, you also start to get false positives in there. So.
Have you tested your fragments against each other as baits and preys to look for within proteins to see if like the domains interact? You know.
We have not done that in a systematic way, but in practice we have, because when we screen with a bait fragment, and then we will pull out prey fragments. So yes, we have many of these interactions that mediate, where we know that two small domains will mediate the interaction. All right, thank you.